Anna, glycolysis is so fermentation is just a pathway that allows glycolysis to continue. Um, and if you remember, excuse me, if you remember, we're going to start with a molecule of glucose, and once that's been transported into the cell, we're going to phosphorylate it twice. It's going to split, and that represents the end of the energy investment phase for glycolysis. And so what we have here are two molecules of G3P, and the cell can now uh, enter the energy extraction phase of glycolysis. And in order to do that, the cell needs to have a healthy supply of NAD plus. Um, this is basically it's a little dinucleotide, two little nucleotides stuck together, and it's got an A base, and then it's got instead of a base like A, T, G, or C, it's got something called a nicotinamide structure. But basically, this small little molecule is going to swoop in, and it's going to rip high energy electrons away from G3P, and so it's going to oxidize it, is what we call it. But oxidizing just means removing electrons, and in the process of oxidizing G3P, it's going to extract a hydrogen as well, so it gets converted into NADH. So that's going to happen to the G3P up top, it's going to happen to the G3P on the bottom, and throughout this process, or as a result of this process, a little pop of energy is released when those electrons are ripped away, and that little pop of energy is used to add free floating phosphates onto G3P. So these, I guess I'll do them in a different color. These phosphates on the left in red, they did not come directly from ATP. They were free floating, and there needs to be some sort of energy source in order to attach them onto G3P. And so that energy source was the energy that was released when NAD plus oxidizes G3P. Now that this has a total of 4 phosphates available, the cell can extract those phosphates off of the G3P and add them onto ADP. And so it does that 4 times because there's 4 phosphates. So it takes an ADP and it sort of recharges it into ATP thanks to these 4 phosphates here. And what you're left with is pyruvate, 2 molecules of pyruvate, and 4 molecules of ATP, and 2 molecules of NADH. Now, this pyruvate, in the presence of oxygen, is going to be sent into the mitochondrion, where it can go through the Krebs cycle, and oxidative phosphorylation can happen, and all that stuff. This NADH is normally going to go off to the mitochondria as well to drop their electrons off into the electron transport chain. But if the electron transport chain isn't functioning because there's a lack of oxygen, or maybe because there's a drug that's blocking the electron transport chain, this pyruvate is not going to be broken down. It's not going to be sent into the mitochondrion because the mitochondrion has shut down, the electron transport chain is shut down. And the NADH isn't going to go into the mitochondrion because, again, the electron transport chain has been shut down. So now the cell has a problem. The problem is that it cannot completely break down pyruvate and get something like, around 30, like I think the textbook average is like 34 ATP can come from the complete breakdown of pyruvate. And also, it's going to start running out of NAD+, because when this NADH goes to the mitochondrion and drops off its electrons, it comes back as NAD+, and so it gets recycled into NAD+, so it can go off and help even more glycolysis happen. But since the NADH is like in a traffic jam, it can't go into the mitochondrion, it starts accumulating, there's a lack of NAD+, and eventually, if all of that NAD+ runs out, this process shuts down as well. So it's like a domino effect. And if the cell is in a situation where it cannot synthesize ATP, it's going to die. And so fermentation is a way to ensure that if this stuff over here is not happening, at least glycolysis can continue. Because what happens in fermentation is all this NADH that gets made, in order to prevent, or in order to not run out of NAD+, the NADH is just going to dump its electrons onto pyruvate. And by doing that, it can turn back into NAD+ and help glycolysis keep happening. The pyruvate is going to get converted into lactic acid once it gets those electrons. And that lactic acid can't be used for anything really. It's a byproduct of this process. So a big mistake that students make is that they say that fermentation is a way to generate energy, but it's not. The only thing fermentation is accomplishing is it's allowing glycolysis to keep happening. All right, let's look at the polar bear FRQ. Okay, so you're looking at amino acid sequence differences. Give me 1 second, actually. Okay, so amino acid sequence differences among these lineages. We have, so polar bear has 40 differences when you compare it to the panda. The brown bear has 34 differences compared to the panda, and the black bear has 33 differences compared to the panda. So if we're going to put our polar bear all the way out here, I think this is what your template cladogram looked like. If you want to put your polar bear all the way out here, what you're saying is that the in group, which is going to be this group right here, the in group is just everything except the out group. So our polar bear would be the out group. You'd be saying that the in group is more closely related to each other. So any member within the in group is more closely related to other members within the in group than they are to the lineage on the out group. So if we wanted to put like our panda right here, and then let me make this smaller, and then brown and black. By doing that, you're saying that the panda is more closely related to the brown and black bear than it is to the polar bear. Right? But the polar bear only has 7 and 8 differences compared to the black and brown, but the panda has 33 and 34 differences. Oh, okay, hang on. I meant from left to right: panda, black bear, brown bear. Oh, okay, sorry. So you agree that panda goes on the outside, which I think is a good placement, right? Because it's very, very different. So panda, left to right: panda, black, brown, polar. Whoops. Okay, so since these are branching off the same node, you're saying that they're more closely related to each other. They share a more recent common ancestor. So if you think of this as like a river or a tree, you've got some sort of ancestral, let me see if I can do this. You've got some sort of ancestral population right here of like 1,000 bears, ancestral bears, like 325,000 years ago or whatever. And that population fragmented. You had a fragmentation, And so we're traveling through time. That fragmentation would eventually lead to the evolution of the panda lineages, right? And then you have another fragmentation. So we're going from past towards the bottom to present towards the top. And this fragmentation would eventually lead to the evolution of the black bear lineage. And then finally, in very recent times, right, so let's say this point right here is current day. Very recently, you had a fragmentation that resulted in the evolution of the brown and the polar lineages. So that's what this node right here is saying. I'll do it in this color right here. This node is saying this right here is the ancestral population of bears that would split and lead to the evolution of these different lineages. And so since this split happened more recently, not too far in the past, there should be very high similarities between these two lineages. But if you look at their similarities between, where's my little scroller? Between brown and polar, what do you see? Brown, polar, you see 8 differences, but you only see 1 difference between brown and black. So that suggests that the brown and the black split, that's the split that happened most recently. And so, well, I keep losing my scroller, right? So that's the split that happened most recently. So it's just based off of this data alone, it makes more sense to argue that this ancestral population doesn't belong to brown and polar. It belongs to brown and black lineages. And we're going to put polar here, right? Because the polar shares an ancestral population with brown and black. That ancestral population is right here, and it's split further back in time. So this is the difference in years, right? So the brown and the black lineages had all of this time within this population right here to sort of evolve together. So there's no differences as long as they're sort of on the same evolutionary trajectory. Those differences only start accumulating after the split occurs. So this is time that the brown and black lineages had to stay the same genetically, that they didn't have with the polar lineage. And so since more time has passed, since those genetic differences were allowed to begin accumulating between those lineages, we would expect to see more genetic differences. And that's what we see. We see more genetic differences. We see 7 and 8. Is that making sense? And the cool thing is that I'm looking at some COVID-19 stuff. I really wish we could talk about this, but it's just, I think it's too much right now. But more different than pandas. No, we can't say that. Okay, hang on. All right. I'll get to your question. Let's see. Uh-oh, what happened here? Okay. That makes more sense, but shouldn't that mean that brown and black should be more different than pandas? Brown and black should be more different than pandas. No, not necessarily. Look, because if you look at the brown and black and you compare it to the position of polar right next to panda is arbitrary. So let me draw the same cladogram, just in different ways. I'll put panda here, and then I'll put brown and black right here, and then I'll put polar right here. Or you could draw the exact same cladogram and put panda here, and put polar here, and put brown here, and put black here. These two cladograms are saying the exact same thing. Or, I mean, you can even make it even weirder. Can we do this? Can we put panda here? Whoops. We can put panda here. We can put brown and black here. And we can put polar here. Right? This is a weird way of doing it, but all of these things are saying the same thing. So the fact that I've got, in this example, the fact that I've got polar next to panda doesn't say anything about the degree of differences between them. We can't say, like, if I'm looking at this group right here, I can't determine or say anything about where there's going to be more differences within any one of these lineages compared to the panda, because their position relative to the panda is arbitrary. You can shuffle it, you can flip it and rotate it, and you're saying the same thing. So the only thing that we can say is that we expect the differences between these lineages and the panda lineages to be approximately the same. Now, obviously they're not exactly the same, and why they're different, that's for further exploration. We don't know that just by looking at this data. But this could just as easily have polar saying 33 and brown saying 40. No, and that's a really common misconception. Left and right isn't giving you that information. It's really reading it from bottom to top. This is time. So no matter how you flip it, like the split here occurred the furthest back in time between the panda lineages and all of the other lineage. That's true for here, and that's true for here, right? So the vertical position of the node is what's important when you're building your trees. Or if you're building your tree from left to right, then the left-right matters. Like if I want to build the tree like this, so brown, black, panda, and then, I'm sorry, polar, and then panda. Then yes, left to right is important because in this case, left to right is time. And in this case, the vertical position doesn't matter. Yeah, the panda is the outgroup, say it, because the outgroup by definition is the group that is least related to all of the other lineages. So the panda is the least related because it has the most differences. So let's see. You can think of these cladograms as like little rivers of time, and so the river right here is just the population of ancestral bears, and each split just represents something that happened that caused that population to fragment. It could be some members of the population migrated away. It could be some sort of geographical event that suddenly separated the populations.